Good evening. Today, the topic of discussion is going to be cell as living units. The human body is composed of large number of cells. In fact, cell happens to be the functional, simple functional unit of our human body. And many cells join together to form an organ. These organs are held together by connective tissue, integuments, all the supporting tissue. And each type of cell is perfectly adapted to perform one or few functions, specific functions. But many cells of the body often differ markedly from one another, but they have certain basic characteristics. These basic characteristics include oxygen inside the cell react with carbohydrate, fat, and protein, releases energy for the sustenance of the cell. And almost all the cells have the ability to reproduce cells of their own type, except for most advanced neural cells like central nervous system cells, where the reproduction does not happen, and any cell that dies in the brain gets replaced by scar tissue, fibrous tissue. So, the more advanced evolutionarily a cell is, less becomes its reproductive or replication capacity. Because as we ascend the ladder of evolution, we have a tendency to reduce the load which we carry. We leave behind these unnecessary things, but even though it is unnecessary for nature, the problem is if there is cell death in the brain, the harsh reality is it is not going to reproduce. It is going to be replaced by fibrous tissue. And cell is supposed to be the basic living unit of the body. And each organ, as I've already told, the aggregation of different cells, they are held together by supporting structures, which could be fibrous structure, integuments, other supporting structures. And these cells number, the commonest cell in the human body happens to be the red blood cell. They number around 25 trillion. 25 trillion, see the number. And let's be the most abundant when compared to any other type of cell. In fact, the entire human body contains 100 billion cells. All cells need energy for them to, and microorganisms normally reside inside the human body and they outnumber the human cells. And these microorganisms has a huge beneficial role to play. Microorganism present in the gut helps in digestion. Now let us talk about extracellular fluid. 60% of the body is composed of fluid, which is a mixture actually. It's a mixture of water, ion, and other substances. And this also contains glucose, fat, and proteins mixed inside the fluid. And majority of the fluid happens to lie within the cell. It is known as intracellular fluid. Roughly about 2/3 of the fluid lies within the cell, that is intracellular, and remaining 1/3 fluid lies outside the cell, that is extracellular. One should remember that extracellular fluid is transported rapidly inside the human body by circulation. within the blood vessels. Cells are actually capable of living, performing their specific function as long as a proper concentration of oxygen, glucose, different ions, amino acids, and fatty substances, and other constituents are available in the internal environment. Extracellular fluid is also called a milieu interior. This term was coined by Claude Bernard. Now, there is a difference in the concentration between extracellular and intracellular fluid. The extracellular fluid contains large amounts of sodium chloride and bicarbonate plus nutrients for the cells such as oxygen, glucose, fatty acids, and amino acids. It also contains carbon dioxide that is being transported from the cells to the lungs to be excreted, breathed out rather, plus the cellular waste products that are being transported to the kidney for excretion. These are all found in the extracellular fluid. The intracellular fluid contains large amounts of potassium ions, magnesium, and phosphate ions instead of sodium and chloride ions, which is found in excess in extracellular fluid. Special mechanisms are available to transport ions through the cell membranes to maintain a concentration difference between the extracellular and intracellular fluid. Powerful control systems are available in the body for maintaining the concentration of sodium and potassium ions. Now, let us come to the hemostatic mechanism. Disease hemostasis, actually, the term hemostasis was coined by Walter Cannon in 1929, and this mechanism ensures that the constant internal environment is maintained throughout the. For example, lungs ensure uniform oxygenation. Kidneys maintain the ionic environment. Various ions, nutrients, and waste products are all concentration of them are regulated within a narrow range. And disease invariably is considered to disrupt homeostasis. Even in the presence of disease, our body homeostatic mechanism continue to operate and maintain the vital function through various methods of compensation. In some cases, this compensatory mechanism could lead to major deviation in the body function from normal range, making it difficult to distinguish the primary cause of the disease from the compensatory response. For example, diseases that impair the kidney's ability to excrete salt and water may lead to increased blood. And this increased blood pressure, what happens? Improves the vascularity of the kidney, and thereby the kidney gets flushed with blood and starts to improve the urine output. The improved urine output provides a route for excretion of salt and water. This is a critical balance that is needed to maintain life over a long period of time. A high blood pressure, on the contrary, can damage various organs, which includes even the kidneys, causing even greater rise of blood pressure and more renal damage. So, homeostatic compensation that ensue after injury or disease or major environmental challenge to the body could represent a trade-off that is necessary to maintain vital body functions. But in the long term, pathophysiology takes over. It should be stressed that even in the presence of disease, homeostatic mechanisms operate to keep the vital functions normally active. Now let us come to the extracellular transport mechanism. Extracellular fluid is transported in two stages. The first stage is actually movement of blood through the body inside the blood vessel. The second stage happens to be the movement of fluid between the capillaries and intracellular spaces between the cells. The blood, in fact, passes through the capillaries with continuing exchange of extracellular fluid occurs between the plasma portion of the blood and the interstitial fluid that fills the intracellular spaces. The capillary walls happens to be permeable to most molecules in the blood plasma with the exception of plasma proteins, because plasma proteins are held back within the capillaries because the pores inside the capillaries are too small for these plasma proteins to pass through. This process of diffusion between the capillaries and the extracellular fluid is caused by kinetic motion of the molecules inside the plasma and interstitial fluids. Now here you see the diagram of blood flow circulatory system. Here, left heart pumps oxygenated blood to the gut where nutrients which are absorbed from the food consumed gets into the circulation, it travels to the therein it carbon dioxide from the gut also gets into blood system, venous system. Then it gets into the right heart, which pumps the blood into the lungs, which manages to extract carbon dioxide from the venous blood that has come to the right side of the heart, and then it just gets into the left heart to be pumped again. The left heart again pumping blood gets into the kidney, wherein oxygenated blood gets into the kidney, supplies the most needed oxygen nutrients to the kidney, and it takes away excess sodium, potassium, and water from the kidney, excreted by the kidney, and gets out. Here again, when it reaches the lungs, carbon dioxide is removed and it is breathed out. So, both these venous and arterial system join together in the capillaries. Capillaries are supposed to have two ends. One is supposed to be the arterial end, the other is supposed to be the venous end. The diagram, the right side you see is the arteriole and venule, and blood and the intracellular fluid. That is, you see the arrows, black arrows, which shows the material exchanges between the extracellular space and within the capillaries. Now, let us see the origin of nutrients, where exactly the nutrients originate in the extracellular fluid. The respiratory system, each time the blood passes through the body, it also flows through the lungs. The blood picks up the oxygen. Oxygen is picked up from the alveoli, and this provides oxygen, much needed oxygen, to the cells. The membrane between the alveoli and the lumen of the pulmonary capillaries are very thin. It is around 0.4 to 2 micrometers thick. The oxygen rapidly diffuses by the molecular motion into the capillaries. Now, gastrointestinal tract, large portion of the blood pumped by the heart passes through the walls of the gastrointestinal tract. Here, the dissolved nutrients and amino acids, fatty acids, carbohydrates are absorbed and ingested into extracellular fluid. The liver and other organs perform a primary metabolic function. Not all substances from the gastrointestinal tract can be readily used by the cells. The liver needs to change the chemical composition of many of these substances to make it more usable by the cell. Now comes musculoskeletal system. Before I go to the musculoskeletal system, I need to talk about another important function of the liver. Liver happens to detoxify blood. It removes ingested drugs, because currently we are exposed to more and more of drugs and chemicals and these drugs and chemicals are removed or detoxified by liver. It's actually a huge detoxification center wherein all the ingested drugs and chemicals are removed. Now, let us come to the musculoskeletal system body. How exactly the musculoskeletal system contribute to the homeostasis? Answer is very obvious. Not for the muscles, the body will not be able to acquire food. The musculoskeletal system also provides the motility for protection against adverse surroundings. It just helps the organism to survive. Now, regulatory body functions include, these are the regulatory body functions: the nervous system, hormone system, protection of the body, that is immune system, integumentary system, reproduction. The nervous system has three parts: the sensory input, the central integration, that is the brain, and the motor output. And there is also in added autonomic nervous system, which controls many functions of the internal organs. And autonomic nervous system is involuntary in nature, is not under higher cortical center control. Hormone system, it secretes hormones. Hormones are transported in the extracellular fluid to other parts of the body to help in regulation of cellular function. The protection of the body is taken care of by the immune system. Then comes the integumentary. Integumentary system includes skin and appendages, which are important for temperature regulation, excretion of waste via sweat. Sweat is again a very potent way wherein wastes are excreted. And reproductive system maintains homeostasis by generating new beings. It keeps the progeny going. Now, control system. The human body has thousands of control systems. Some of them are most intricate, and many of the control systems operate within the cell, intracellular control system. And some of the control systems operate within the organ, at the organ level, that is organ level control system. And then there are some control system which controls the interaction between various organs. So interrelationship, they control the interrelationship between the various organs. Now, let us see what are all the various control systems. The first one we are going to see is the nervous system, which uses electrical impulses and neurotransmitters to regulate rapid and short-term responses. Central nervous system or nervous system per se has rapid response, creates rapid response, whereas endocrine system uses chemical messengers which are secreted. This slow action is rather slow, long term. So long term process like growth, metabolism, and reproduction are dependent on the endocrine system control. Homeostatic mechanism, they involve feedback loops. Common feedback loop that is used by the human body to maintain homeostasis is negative feedback mechanism. For example, maintenance of the normal temperature of the body, maintenance of blood glucose level, these things are taken over by the homeostatic mechanism. Then the genetic control system. This is actually the intracellular control system which operates within the cell, thereby directing cellular activity. And also this also interacts with other control systems. Now, I will narrate some of the examples of control system mechanisms. Regulation of oxygen and carbon dioxide concentration in the extracellular fluid. Because oxygen is one of the major substances required for chemical reactions in the cell. The body has special control mechanism to maintain almost the exact concentration of oxygen in the extracellular fluid. This mechanism depends principally on the chemical characteristics of hemoglobin. which happens to be present in all the red blood cells. Hemoglobin combines with oxygen as the blood passes through the lungs. Then, when the blood passes through tissue capillaries, hemoglobin, because of its strong chemical affinity for oxygen, does not release oxygen in the tissue fluid if there are too much oxygen is already there. However, if the oxygen concentration in the tissue fluid is low, sufficient oxygen is released to re-establish the adequate concentration. This regulation of oxygen concentration in the tissues relies to a great extent on the chemical characteristics of hemoglobin. This regulation is known as oxygen buffering function of hemoglobin. Similarly, carbon dioxide concentration in the extracellular fluid is a tightly regulated mechanism. Carbon dioxide, in fact, is a major end product of all the oxidative reactions inside the cell. If all the carbon dioxide formed in the cells continue to accumulate in the tissue fluid, all the energy giving reactions of the cell would cease to function. Fortunately, higher and normal carbon dioxide concentration in the blood excites the respiratory center. The respiratory center, in the presence of increased level of carbon dioxide in the extracellular fluid, causes an individual to breathe rapidly and deeply. Rapid breathing increases the expiration of carbon dioxide and therefore removes excess carbon dioxide from blood and tissue fluid. This process continues till the concentration of carbon dioxide becomes normal. Then another important example I would like to stress is regulation of arterial. Several systems contribute to arterial blood pressure regulation. One of these happens to be the baroreceptor system. In the walls of the bifurcation of the carotid artery in the neck as well as in the arch of aorta, there are receptors that gets excited or stimulated when they get stretched by the increased pressure. So when arterial pressure rises too high, the baroreceptors send impulses to the medulla of the brain. Here, the impulses inhibit the vasomotor system, which in turn decreases the number of impulses transmitted from this center through the sympathetic nervous system into the heart and blood vessels. So this causes reduced pumping activity of the heart and dilatation of the peripheral blood vessels. Lack of these impulses and diminishing pumping activity causes increase in the blood pressure. If decrease in arterial pressure occurs below the normal, the stretched receptors gets active and thereby causes vasoconstriction and increases the pumping rate of the heart. So thereby it improves the blood pressure. Now let us discuss the various mechanisms involved in genetic control. The first mechanism of genetic control is gene expression and regulation. Here, this control mechanism is involved in controlling the expression of gene as well as it regulates the gene expression. Then the second one happens to be epigenetic regulation. The third one happens to be regulatory sequences and transcription factors. Fourth happens to be non-coding. Now, let us discuss about gene expression and regulation. Transcription control, how gene expression works, controls how often a gene is transcribed from DNA to messenger RNA. So, the details from the DNA has to be matched or transcribed to messenger RNA. Here, this transcription control has the ability to turn a gene on or off to regulate RNA production. For this purpose, RNA polymerase enzyme is used. Then next comes RNA processing control. This RNA processing control mechanism regulates how RNA transcripts are spliced and processed. Then RNA transport and localization. This control system determines which messenger RNA are exported to the cytoplasm and other cellular destinations. So RNA transport and localization is one very important control system that really determines which messenger RNA is exported to the cytoplasm and other cellular destination. Then translational control mechanism, wherein the messenger RNAs are translated into proteins. Messenger RNAs are those RNAs that gets translated into proteins. So this control ensures that tight control is exercised over the translation of proteins. Then messenger RNA degradation control. The third is protein activity control, which selectively activates and inactivates and degrades. Now let us come to epigenetic regulation. Chemical modification of DNA. For example, addition of methyl molecule to DNA or methylation on histones alter the gene activity without actually changing DNA sequence. Histones are actually should be are 4 types: H1, H2, H3, H4. These are supposed to be packaging material that packages the genes. So histones and other methylated DNA can alter, because some packages can be round, some packages can be square, the shape depending on the shape of the package. So similarly, when the gene is packed and different types of histone, modification occurs without actually the inner sequence of the DNA does not change, but the covering, packaging of histones changes its form. This epigenetic regulation enables dynamic and reversible of the gene activity. This particular regulatory mechanism enables dynamic, number one, control on the go, and then the control can be reversed over the gene activity. It influences development of the cell, differentiation of the cell, and adaptation of the cell. Now, regulatory sequence and transcription factor controls. Here, there are promoters, enhancers in DNA that serve as binding sites for various types of proteins. These are called actually transcription factors. They activate or suppress the gene transcription, ensuring a tissue specific. I repeat, this control is exercised in a tissue specific manner and condition specific gene expression. Tissue specific and condition specific gene expression. Now let us come to non-coding RNAs. These non-coding RNAs are long microRNAs, long non-coding RNAs. They can bind to the messenger RNA, regulate the messenger RNA, and affecting whether, how, if at all gene product is going to be made, how these products are going to be. So this control is exercised by microRNAs, long non-coding RNAs, because they have a direct influence on the messenger RNA. Now, let us get into the nitty-gritty of the functional role of the genetic control. Every cell, should be remembered, has the same genetic material. The genetic material inside each cell are absolutely identical, but the expression of certain genes are needed for this function. Even though the genetic material residing inside each cell are the same, but expression of certain genes as needed for function, like liver cells needs certain genes to be preferentially expressed. Similarly, skin, muscle cells, and these specific activation of these genes, special genes, helps in the differentiation of cells. So genetic control system are responsible for cellular differentiation, functional tissue, and then functional tissue function, development of the organism, as well as dynamic response, which happens due to external and internal stimuli. Now, let us dwell a little bit into non-coding RNAs. There are 4 types of non-coding. MicroRNAs, they regulate gene expression by binding to the messenger RNA. All these microRNAs, they regulate gene expression by binding to the messenger RNA. Then there's something called transfer RNA. These are all non-coding RNA. They don't exactly have code for any gene. So these transfer RNAs carry amino acids to the ribosome. Ribosome is actually the protein factory, protein making factory within the cell. So they transfer RNAs has a tendency to transfer amino acid to the ribosome during the protein synthesis, enabling protein synthesis. And ribosomal RNA forms is capable of forming the structural and catalytic core of ribosomes. Ribosomes core structure is taken care of by ribosomal RNA. This RNA forms the core of the ribosomes. So the ribosomes are those organelles that are capable of protein synthesis. Small nuclear RNAs, they guide the chemical modification of other RNAs. Small nuclear RNAs, they guide the chemical modification of other RNA. Now let us come to negative feedback and positive feedback system. So to maintain homeostasis, the body depends on feedback mechanism. The commonly used feedback mechanism that the body depends on to maintain homeostasis happens to be the negative feedback mechanism. Negative feedback systems maintain stability by counteracting a change to return to a set point. Whereas positive feedback system amplify the initial change to push a process to completion. But negative feedback is the most common for maintaining homeostasis, such as regulating body temperature, blood sugar, whereas positive feedback mechanism is seen in specific situations like childbirth and blood coagulation. So, as I've already stressed, most of the control system of the body act by negative feedback mechanism. And this in regulation, this can be aptly seen in regulation of carbon dioxide concentration. A high concentration of carbon dioxide in the extracellular fluid increases pulmonary ventilation. This in turn decreases the extracellular carbon dioxide concentration because lungs expire greater amounts of carbon dioxide from the body. Thus, higher concentration of carbon dioxide initiate events that decrease the concentration of carbon dioxide. So, this is the reason why this is called negative feedback mechanism, which is the value gets reduced, that is negative to the initiating stimulus. In the arterial pressure regulating mechanism, a high pressure causes series of reactions that promotes reduced blood pressure. The aim of this control mechanism is to reduce the blood pressure. Hence, it is a negative feedback mechanism. Now, these are some of the classic examples of negative feedback mechanism. Thermoregulation. If the body temperature rises, body starts to sweat to cool down. If it falls, if the body temperature reduces, the body shivers to warm up. Then blood glucose regulation. After a meal, what happens? The sugar rises, the body releases insulin to keep the sugar level under check. Blood pressure regulation. If the pressure rises, heart rate decreases to lower the blood pressure. Now, let us look into some of the examples where positive feedback is involved. Here, childbirth, the classic example. Baby's head, when it presses the cervix, it releases oxytocin. This causes uterine contractions to become stronger. This pressure and oxytocin release, continues to amplify concentration until the body is born or the baby is born. So the pressure and oxytocin release continues and amplifies the concentration until the baby is born. Then blood clotting. When a blood vessel is damaged, platelets gather to the site, release chemicals, attracting more and more platelets, causing the clotting process to start. Now, let us discuss about the gain control system. Gain of the control system. The degree of effectiveness. I repeat, the degree of effectiveness with which a control system maintains constant condition is determined by the gain of the negative feedback. For example, let us assume large volume of blood is transfused into a person, but the baroreceptor pressure control system is not working in that individual. So what happens is the arterial pressure rises to from 100 to 175 millimeters of mercury. Again, if you assume same volume of blood is transfused and the baroreceptor system is functioning, the pressure increases by only 25 millimeters of mercury. Thus, the feedback control system, actually in term you can call it as correction, happens around 50 millimeters of mercury. It reduces by minus 50 millimeters of mercury. So 170 from 175 millimeters of mercury, the control system reduces the blood pressure, arterial pressure to 125 millimeters of mercury. So there is a notional increase of just plus 25 millimeters of mercury. This number, this value is in fact called as the error. This value is known as the error, which means all the control systems, whichever it could be, is not 100% effective or foolproof. So gain of the control system is calculated by this formula. Gain is equal to correction divided by error. Thus, in the scenario I have explained, the correction happens to be minus 50 millimeters of mercury. On the error, the system has not reduced the BP or arterial pressure 100%. So there is it has reduced the arterial pressure by just 50 millimeters of mercury. So the effectively current arterial pressure happens to be 125 millimeters of mercury. So what happens is the error happens to be plus 25 millimeters of mercury. So if you use this formula, you get a value of minus 2. So you just have a look at this formula. You will get in your competitive examination, NEET and all that, you may have to apply this formula to get at the gain value of a control. They will give you a control system value and they will ask you to calculate the gain. Unless and until you know this formula, you won't be able to calculate the gain value of a control. Thank you so much.